So I just want to take a quick moment to introduce the chair for the next session. This is particularly meaningful to me because Dr. David Jaffrey is literally the person that introduced me to the journey into theranostics over 10 years ago. And together with Dr. Mary Gaspadarowicz, sowed the seed of what UHN is doing and building contributing to this big network in front of us today. Dr. Jaffrey is a visionary. He was talking about nanoparticles and molecular targeting way before it is cool. He's currently the Senior Vice President, Chief Technology and Digital Officer, full professor at the University of Texas, MD Anderson Cancer Center. But before that, he was here in Toronto. He spent 17 years at UHN, Princess Margaret, and had countless accomplishments and legacy. He founded multiple innovative centers, institutes that supported innovation, including STAR and TECNA. His inventions and writings has translated into new technologies that we use every day today. His advocacy has improved cancer care on a global setting, especially in low and middle income countries. He's contributed to the careers of countless physicists, oncologists, therapists, and scientists. So I'll hand over to Dr. David Jaffrey.
So good morning, everybody. It's great to be back up here in Toronto, and it's super exciting to see and hear the energy, the nuclear energy that's going on in this audience. So I'm delighted to be the chair for the session and delighted to introduce Dr. François Bénard. François is a BC Leadership Chair in Functional Cancer Imaging. He's based at University of British Columbia. He's a professor in the Department of Radiology. He's also the Associate Dean. He's also Vice Principal Research and Distinguished Scientist at BC Cancer. In 2015, he received the NSERC Brockhaus Canada Prize, and that's a prize dedicated to transdisciplinary and interdisciplinary science, fusing engineering with medicine. Now, I have a little bit of history with François. We've been exchanging emails for multiple decades, actually. And he was in Sherbrooke, and he had just gone to University of Pennsylvania for a fellowship in nuclear medicine. He came back to Sherbrooke, and we heard about this young talent. And so I was in Toronto. We were starting to build a program in molecular and functional imaging that grew into theranostics. And so we tried really, really hard to recruit François to Toronto. And we pulled so hard that he went right past us and went to Vancouver, okay? And then in 2010, we tried again. We pulled back, and we had a huge crew. Ben Neal, Chris Page, Tom Hudson, many others tried to pull him back, but he is solid at BC. So we're going to hear about his work on optimizing diagnostics and therapeutics in the radioisotope space, and I'm looking forward to his presentation. Hopefully we'll have a few minutes for questions. I'll give you Dr. Bénard.
Thank you, David. I'm going to put my timer just to make sure I stay on time and we have a bit of time for questions. I'm going to talk today not about how to develop a new molecule that will target a specific protein on cancer cells, but once you have this targeting vector, how do you go about in turning this into a potent diagnostic or therapeutic radiopharmaceutical? First of all, I just start with my conflict of interest disclosure. So I'm a co-inventor or co-inventor of multiple patent applications. I am a co-founder, a shareholder, and a consultant working with Alpha-9 Oncology, which is a startup company. It was founded by UBC and BC Cancer out in Vancouver, and now the company is run fully independently. I also have some intellectual property, and one of the co-inventors, which led to the Brockhaus Award, and led to the Artemis being founded, although I never was involved in the company. Now, we all know about theranostic. The basic principle is we have a specific cancer target, so a protein that's selectively expressed or overexpressed on a cancer cell. Usually it's an imaging test that will decide whether or not we can use this for treatment. And if the imaging test shows that there's high or consistent expression of the target, then we can treat this patient. If there's inconsistent expression or low expression, then those patients are probably better treated by other means. So developing a diagnostic companion is extremely important, not only for as a simple yes or no diagnostic answer, but also to look at expression heterogeneity, because cancer is a very complex disease and it evolves over time. Often we see patients who've had multiple rounds of treatment when they get to receive radioligand therapy. So these treatments drive clonal evolution of cancers who become more heterogeneous as they evolved. And we see this, for example, in the case of advanced metastatic prostate cancers. By the time they receive Pluvicto, for example, if the disease sufficiently advanced, about 20–30% of patients will have evolved into developing what's called a neuroendocrine prostate cancer differentiation, which loses PSMA expression, expresses new biomarkers, but becomes very, very, very challenging to treat in a rapidly progressive disease. So we have a number of choices when we're developing radioligands. This is a conference that's focused a lot on isotopes. So there's radioisotopes for diagnosis, including technetium-99, which is widely used in nuclear medicine, electron capture isotopes, iodine-123, where Canada is one of the globally leading producers, and positron emitters, such as fluorine-18, gallium-68, and so on. The therapeutic isotopes are broadly in three categories: beta emitters, which emit light particles that carry a slightly longer distance and will cause mostly indirect DNA damage. Auger electrons and some isotopes that emit conversion electrons, so low energy particles that emit particles at a very short range. So these often need to be very close to the nucleus to cause DNA damage. And alpha emitters, which are like bowling balls that don't go very far but cause a lot of damage when you throw them, and a higher proportion of double strand breaks. So even in the space of using radioisotopes for therapy, we've got different mechanisms of action. Some isotopes will give predominantly indirect DNA damage, causing free radicals, and that can lead to cancers developing ways to evade this, for example, by ramping up their antioxidant capacity, or alpha emitters, which seem to be more potent and can overcome resistance to other forms of radiation treatments. Once you've developed a therapeutic a molecule that binds to a cancer cell, you have to think about how are we going to get this into the clinic, and it still is a very challenging problem. If you can use technetium, that's fairly simple because it's widely available globally. But if you want to use PET tracers for greater resolution, better detection, better quantification, then there's three mainstream choices currently on the market. F18 is probably the best isotopes for diagnostic purposes. Gives great image quality, favorable dosimetry, widely available because of the FDG availability. However, for countries like Canada, the distribution is much more challenging. In the US, there's these large networks of well-established commercial radiopharmacies where you can make a partnership with a company like Cardinal Health or PetNet, and then you immediately have a United States-wide distribution center, and they can make your molecule. It's not cheap, but they will partner and make it available for distribution. In Canada, we have a patchwork of mostly academic, hospital-based or university-based cyclotrons with commercial cyclotrons as well in Ontario and Quebec. So enabling distribution requires a lot of legwork, and there's not been much success so far in establishing that model in Canada, so it does remain a hurdle. Gallium-68 can be distributed. You need to make germanium-68, which is made by high-powered cyclotrons. It can be packaged in a generator. The generator will have typically a shelf life of about 9 months. You can ship them to multiple hospitals. And that model is working well, and we're seeing this for diagnostic kits for neuroendocrine tumors and prostate cancer. But again, there's more barriers because of the cost of generators, implementing this in conventional nuclear medicine departments, which have not used these kinds of generators before. But it certainly is something that works. Gallium-68 can also be produced by cyclotrons in larger quantities, but when you get to that level, F18 is a good alternative as well, and has got a longer half-life. But if you have a kit, for example, like the PSMA kits, a company called Telix is developing also cyclotron-produced versions of this, so you can ramp up production. for regional distributions. Copper-64 is a radioisotope that some of us have worked with. Susie is here. She's had a lot of experience working with copper-64. It's got almost a 13-hour half-life, can be centrally manufactured, distributed elsewhere, provides great image quality for PET imaging. But unfortunately, the dosimetry is less favorable than F-18, but for some tracers that require a little bit more time to achieve peak diagnostic performance, copper-64 is a good alternative. There's also a copper-67 equivalent, which is a great beta emitter that can be used for therapy, and it's got a really good half-life as well that's shorter than lutetium. Now, when you develop the diagnostic radiopharmaceutical, the overall goal is fairly simple, right? We want to maximize the signal, maximize the payload delivery to a tumor cell so that you have a great contrast relative to the adjacent background of the areas where you expect the tumors to be located. Ideally, you want that in a practical time frame. Now, patients don't want to spend their days in hospital. If you have a family member who suffers from cancer, you know that they get tired of going to the hospital. Even 1 day a week is a lot. Often they get called 2, 3, 4 days a week to go to the hospital. So if they have to spend hours sitting there waiting for more tests and so on, it's very cumbersome psychologically and physically for them and for their family members as well. So you want to do this in a relatively quick time frame. 2 hours or less, probably 1 hour is better. But you want low background activity in the areas of interest, high tumor uptake, and you want a minimal radiation exposure. People who've had one cancer are very worried about getting other cancers, and there's people with genetic predispositions, some that are known, others that are not known, that make them susceptible to cancer. If they get high dose of radiation or systemic radiotherapy, you could argue that the dose is minimal, but for people who've had a cancer that's likely to be cured, they are worried about radiation exposure over the long term, so we want to minimize that. And there's other pitfalls, like GI excretion can be quite cumbersome for some if you're looking at gynecological or GI cancers. Urine excretion, if you're looking at bladder cancer, can be a problem. Prostate cancer you can manage, but it still is an issue. And you can also have low uptake when the tracer is excreted way too quickly, or for example, if it's sunken into the kidney or normal organs, liver or spleen, then it can force the compound to clear too fast and there's not enough tumor uptake, for example. Before moving on to I just wanted to go back to the choice of isotope. And this is an example of work I did with me, my trainees, and Dr. David Perrin and Koshian Lin when we have a colleague at UBC called David Perrin who developed new methods for radiolabeling biomolecules. And at the time, it was very complex to attach fluorine-18 to peptides. You needed indirect reactions, label the first molecule, then attach it to a peptide. It was a 2-hour-long process with relatively low yields. And there's 2 competing technologies, or 3 now, for labeling peptides with F-18. This is one example where you have a small moiety called a trifluoroborate, which is labeled 1 on that screen. And we decided to use a simple, well-known peptide called octreotide to use it as a test molecule to label with the F-18 molecule. So in this case, it's different than other ways of radiolabeling. The same molecule is the one that you radiolabel, and you do an exchange reaction to swap one of the cold fluorine with a radioactive fluorine. And this is an example of how you can label molecules in a very simple manner, and we translated that into the clinic. I think the animation is missing, but you can get the gist. This is exactly like what you would see with a gallium octreotide scan, except with higher spatial resolution, better statistics, because it's F-18, about half the dose. Uptake in tumors is very quick. You can scan within 15, 30 minutes and get very stable uptake for 2, 3 hours of the same compound. So these are the kinds of ideal radiopharmaceutical. We can't completely get rid of some of the uptake like pancreas and liver because this is where the target is expressed as well. There's my picture. Another issue to control is metabolic stability. If you work with small molecules or peptides, our system is made to metabolize and destroy unknown substances very quickly. So peptides we use peptides as hormones, but we need to control how long they stay in the blood. So we have lots of enzymes that are geared to destroy these peptides very quickly. some of the peptides that are secreted in our body have a plasma half-life of less than 2 minutes. So there's a series of peptidases, some expressed in the lungs, some expressed in the kidneys, some in the liver, some in the blood, that will metabolize peptides. If you use the small molecules, drugs are metabolized. Everybody who does drug development knows that this is a very important issue to control. And rapid metabolism can also prevent you from developing an effective diagnostic or therapeutic compound. And there's an example with a compound we've been developing over a few years. So this is work that our team has been working on with Dr. Koshan Lin. We labeled bradykinin, which is an acute inflammatory molecule. It's a peptide that's secreted when there's tissue injury. There's 2 receptors for the bradykinin receptor. The bradykinin 1 receptor, which is not normally expressed in our cells, it's only present when there's tissue injury. It will emerge over several hours or days after tissue injury. And the bradykinin 2 receptors, which is expressed in our vessels and will cause immediate vasodilatation, permeation of the blood vessels, and so on. So we're targeting the bradykinin 1 receptor, which has been reported to be expressed in tumors as well as some inflammatory conditions, while avoiding binding to the bradykinin 2 receptor. However, when we were developing those, on the image on the left, you can see we barely saw any signal in the tumor, and this was a cell line that we had transfected with the receptors to express large quantities. But by giving peptidase inhibitors, we were able to see a signal. So this gave us a cue that the problem with poor tumor visualization was not the binding molecule itself, it was just getting metabolized too quickly. So we went on and developed stabilized versions of the other molecule successfully, and these compounds are now in clinical trials ongoing in Australia. Another example is with another compound we developed targeting the chemokine receptor 4. So this is a receptor that's expressed in a large number of cancer types, about 16 or 18 different cancers overexpress the CXCR4. And notably the hematological malignancy, acute myeloid leukemia, multiple myeloma, many types of lymphoma, for example, have very high expression of the CXCR4 receptors. And we used a peptide that had been developed by Lilly as a base targeting molecule and attached a linker and a chelate on it. And when we were trying to explore the effects of the linker, so the first molecule was called BL01. It's my colleague, Dr. Joseph Flau, who started working on this. And if you wonder what the BL is so he called it the Bénard and Lin 01, because he was on his way to the NIH and for some reason decided he wanted to leave this name as a legacy. So you can see that it was good tumor uptake, relatively moderate liver uptake. We were quite pleased with that, right? When this is a first generation compound, you're super happy to see that kind of tumor signal. And then we started playing with the linker to try to see, can we optimize and improve the distribution of this compound? So often we've got a really clear set of ideas of how to go about optimizing this, and playing with the charge properties and the hydrophilicity of these compounds, making them more soluble in water or less soluble in water are some of the strategies we use. So when we put a positive linker on the right, and call it a cationic linker, meaning we put a positive charge on the linker, all of a sudden the tumor uptake was lower, background uptake was higher. Add a couple of negative charges on that, then all of a sudden we had the reverse reaction. We had much better contrast, better tumor uptake. Now every peptide is different, every receptor is different, and it's not only linked to the structure of the protein that it binds to, it's important, but it's also the overall charge and how your body will dispose of a peptide. So it's kind of a tricky set of complex reactions. Eventually, once we have enough data, an AI bot will be able to replace our kind of empirical approaches to doing this. But this is an example of how we can optimize this. So so apologies for the duplicated structure. This is not deliberate. But another one of my students, Daniel Kwan, was making systematic modification of the peptide and replacing every amino acid that he thought could be substituted successfully. So we moved the location of the radioactive payload on the peptide, and we're able to get images like this with extremely high tumor to background ratio, essentially no background and very, very high tumor accumulation. And since we had the technology for radiolabeling with fluorine, then we've been able to make an F-18 compound. an F-18 version of that with extremely high contrast. And now this is in current clinical trials that are ongoing. Another important concept that we've kind of lost sight in our community for a few years is the importance of molar activity. Molar activity is the amount of radioactivity you have per amount of mass of compound injected. So the principle of nuclear medicine is you work in a tracer state. You inject so little molecules that you do not affect the system that you're trying to measure. And this has been true when you work, for example, with very high expressing target. In the context of PSMA, for example, which we use for prostate cancer diagnosis and therapy, we're working with 100 to 500,000 copies per cell expressed in prostate cancer cells. If you're looking at the neuroendocrine tumors, you're tens of thousands of copies per cell. But some of the targets of interest that are emerging have as little as 1,000 copies per cell. And it's been true, for example, with some of the new ligands developed by some companies. It's true with one compound we've been working on called the melanocortin-1 receptor, which is expressed in malignant melanoma. And it's been known for people who do neuroreceptor imaging for many years, right, that you needed to inject extremely small quantities. First of all, you don't want to drive your patients crazy by stimulating the wrong neuroreceptor and adding too much cold compound. But it was known that these low-expressing systems that you want to measure very selectively could get easily saturated if you put too much cold molecules. And it can drastically affect the distribution of a radiopharmaceutical. And this is an image of, this is a graph that shows what happened when we were, this is mouse data where we put a human melanoma model that had very low expression of the melanocortin model, the melanocortin receptor. The percentage injected dose per gram is the amount of tracer that's taken up, the amount of what you're injecting. So 8% is pretty good, 15 is better. 3-4% is generally not optimal, right, for a radiopharmaceutical. But as soon as we injected in a mouse, so the unblocked means about 5 picomoles of mass injected in a mouse. Very, very little. And I suspect that already is starting to saturate. When we injected 20 picomoles, more than 50% of the tumor uptake was down. Now, 20 picomole in a mouse is about the equivalent of 15 micrograms in a human when you scale up from mouse to humans. And when we label a kit with gallium-68, how much do you think we're using in terms of cold molecule in our gallium kit? Anywhere between 20 and 50 micrograms. So 20 micrograms would be a typical kit for labeling with gallium. That means with a compound like that, you're already, if you inject your vial in a patient, you will saturate your tumor receptor. So this is something that's going to become increasingly important as we move from highly overexpressed targets to targets that are selectively expressed but at much lower levels. Even in a compound that has very high expression like FAPI, and this is a recent publication by Liu and colleagues that was published in the European Journal of Nuclear Medicine, you can see the effects of molar activity, and this is in mice, of course, again, but the one image on the left is about slightly less than, is about 5 picomole. That's probably the best we can do with those kinds of tracers. And the image on the right, we are decreasing the molar activity going from left to right. And you can see that the background decreases as we give more cold molecules. The tumor to background improves to some extent, but the tumor uptake also decreases. So this is quite an important parameter to control when doing studies. So I'll have to accelerate quickly because my watch is telling me that it's time. When you're optimizing for therapeutic applications, it's a slightly different problem. Many of the common objectives for diagnostic compound are the same. However, you want the compound to remain longer in the tumor. And you want it's also possible to increase tumor uptake compared to a diagnostic radiopharmaceutical because you want your compound to circulate multiple times. If it got slightly slower clearance, you can put more of your isotope in the tumor. The big advantage is you can use less isotope to be able to get an equivalent therapeutic effect. So for example, if you're able to give 50 millicuries instead of 200 millicuries for a Pluvicto dose because your compound has higher tumor and longer retention, then you would reduce your isotope production costs, or you can treat 4 times as many patients for the same production cost. However, to do that, you need if you slow down the clearance, that means your molecule is more vulnerable to metabolic inactivation. So, for example, with the melanocortin compound, when we put an albumin binder on those, we realized that we did not get any benefit because the molecule was getting degraded sufficiently quickly that any advantage of keeping it in circulation longer was lost. So we developed, Cheng-Cheng Zhang, who's one of our postdoctoral fellows, developed a metabolically stabilized version of this compound. I'll skip this. And the other way, just the way we currently use mostly to slow down the clearance is by adding what's called an albumin binder. So it's a small molecule we add into the peptides to carefully slow down the clearance. And there's a wide range of albumin binders. Most of those that are being used are derived from what's called phenylbutyric acids. And by changing from a fluorine to a methyl to an iodine and bromine, you can control the clearance of that particular compound. Some are faster clearing, others are not clearing. So I'll finish just by showing the work of our student, Itzel, who's been developing improved versions of the CXCR4 binding ligands. And on the left, you see the compound we developed for imaging that she labeled with lutetium to target a different type of cancer that has lower expression than the hematological malignancy. This is looking at neuroendocrine prostate cancer. It's a really, really difficult to treat cancer. And you can see we get good tumor uptake on the images of the panel on the left. At 1 hour, we get 12% injected per dose, but at 4 hours, it's already starting to clear out. This is not an internalized molecule. It stays at the surface and leaches out of the tumor relatively quickly. So this is not optimal for therapy. So what Itzel did is she combined this with an albumin binder. Tumor uptake was improved about threefold. But not only that, because the albumin binders helped internalization, the compound was retained much longer, and you end up having several days later, you still have 11% injected dose per gram, and while still keeping the background low. So these are some of the examples of how we can optimize payload delivery and turn something that probably would not have worked into a therapeutic to become much more potent. We've done the same, for example, with PSMA compounds. On the left is the PSMA 617 molecule. On the right is when we've made specific modifications to optimize, decrease the background, reduce kidney retention, reduce salivary gland accumulation, improve payload delivery. And essentially, we've been able to improve about 5-fold the maximum tumor uptake, much slower elimination from the tumor, so the optimal dose delivery was about 10-fold. So I think I'll stop here. Just to finish is that mice are not humans. So the next step after you develop and optimize for mice is it needs to get into clinical trials quickly. And that means being able to do imaging trials in a relatively quick time frame and sometimes having to compare multiple compounds in humans. And it's probably a nice segue in the next panel is that early phase human studies are critical in the next phases of development.
I want to compliment François. I mean, to go from modes of decay all the way through to the latest science and optimization. I'm going to ask you one question because it would be a shame not to let the audience hear. So you said optimization like 150 times. What is the opportunity from a computing perspective? Not just the AI and the folding of the molecule, but to really bring all these different parts together, maybe in like a fully computational model to optimize. Just a few quick response before we go to the panel.
Yeah, so there's from the molecular docking is becoming increasingly important. You've got AlphaFold that can predict the structure of a protein. You've got very powerful computing models that can be used to model the docking. That part is getting better, but very small changes in the molecules that the models would say will work well or something are not predicted well by those models yet. So we need a lot more data on every system to be able to develop predictive models that are more accurate. The in vivo pharmacokinetic parameters, I do agree, this is ripe for enhancement and prediction, looking at the pharmacokinetic modeling, and my colleague Arman Ramim is doing some more early work on that and trying to see if we can predict the optimal range, the optimal clearance rate, the first pass extraction, and predict which is the best approach. And right now we're optimizing in mice, which is problematic because humans will behave differently. So the risk is picking the wrong lead candidate that might be the best one in a mouse, but will not be the best one in a patient. And even non-human primates will not predict that. So I think we have to get to a stage where we get more and more data. We can develop those predictive models, but we also need to be able to have a regulatory framework that allow us to run a panel of diagnostic tracers so we can see, okay, which one behaves better, and that will help us guide the better therapeutics.
Super. Okay, one more round of applause for our speakers.